Pharmaceutical Dispersion System to Self-Microemulsifying Drug Delivery System SMEDDS and Biomedical Application. Myself, Dr. Prashant Upadhyay, Associate Professor, School of Pharmaceutical Sciences, IFTM University in Moradabad. Introduction. The dispersed system is defined as a system in which one phase, the dispersed phase, is distributed uniformly as particles throughout another phase called dispersion medium or continuous phase. Dispersed system can be classified on the basis of physical state of two phases, the dispersed phase and the dispersion medium. Classification of dispersion system on the basis of particle size. Mainly there are three classes of there. First one is the for the biomedical application. Why we need SEDDS? Crystalline drug is the slow in the may be insufficient time for the process during intestinal transit. Means they are very slow to release. SNDDS helps in the dissolution step, is avoiding and more drug is being available at the site. Composition of SMDDS: oil, surfactant, co-surfactant and polymers. As already discussed, oil are the most important excipient because oil can solubilize the lyophilic drug in specific amount. Both long chain triglyceride, medium chain triglyceride oils with different degree of saturation have been used for the formulation of SMDDS. Unmodified edible oils have poor ability to dissolve large amount of hydrophilic drugs. Modified or hydrolyzed vegetable or edible oils have contributed widely to the success of SMEDDS owing to their formulation and physiological advantages. MCTs were preferred in the earlier self-emulsifying formulations. The absorption enhancement is greater when using unsaturated fatty acids. Examples of oils: corn oil, monoglycerides, corn oil, olive oil, oleic acid, sesame oil, hydrogenated vegetable oils, soybean oils, peanut oil, beeswax. These are certain used, and more fractionated triglyceride coconut oil is being used as oil. Surfactants: natural surfactants have limited ability to emulsify. Non-ionic surfactants are less toxic when compared to ionic surfactants. The usual surfactant strength range between 30 to 60%. Non-ionic surfactants with high hydrophilic lipophilic balance values are used in formulation of SMDDS. Surfactants are amphiphilic in nature. Amphiphilic means both hydrophilic and lipophilic in nature. They can be dissolved or solubilized relatively higher amount of hydrophobic drug compounds. Example of surfactant. So means surfactants are the major constituent in the self microemulsifying drug delivery system. Polysorbate 20, polysorbate 80, twin 80, other name is 20, polysorbate monooleate, span 80, and such as the modern surfactants are there. Lebrafils are there, Cremophore is there, and such so on. What is the co-solvents? Co-solvent may help to dissolve large amount of hydrophilic surfactant or the hydrophobic drug in the lipid phase. When there is a certain solubility problem with the drug, then co-solvent or co-surfactants are used along with the previously existing surfactants. These solvents sometimes play the role as co-surfactant in the microemulsion system. Alcohol is not included in SDDS due to its migrations. Drug release is increased with increasing concentration of co-surfactant in the formulation. Example of co-solvents: ethanol, polypropylene glycol, polyethylene glycol, polyoxyethylene, propylene carbonate, and so on. Polymers, again, they are used, and polymers such as hydroxy propyl methylcellulose HPMC and ethyl cellulose are used in sustained and controlled release of SMEDDS. They are The controlling, or you can say the controlling or sustained release polymers are there, because they release the drug as per the desired rate towards the site. Preparation of SMEDDS. Accurately weighed amount of drug was placed in a glass vials, whose surfactants are added, components are mixed with gentle stirring, vortexing mixing for 30 minutes, mixture is heated at 40 degree centigrade on magnetic stir until drug is perfectly dissolved. The mixture was stored at room temperature until further. Factors affecting acidity: nature of oil, surfactant concentrations, ratios, temperature at which self-emulsification occurs. Drugs which are administered at very high dose are not suitable. Ability of to maintain the drug in solid form is greatly influenced by the solubility of the drug in oil phases. In vitro evaluation of SMDDS. Droplet size analysis, zeta potential measurement are the common, because they have the very low size range droplets. So these are essential measurement. Viscosity determination by group field, in vitro drug release, thermodynamic stability studies, dispersibility test, drug content analysis, turbidimetric evaluation, diffractive index, percent transmittance, electroconductivity studies are being studied. Refractive index, percent transmittance are these. Electroconductivity studies, turbidometric evaluation is done to monitor the growth of emulsification. What is the application of XML? Means what is the biomedical application? Improvement in solubility and bioavailability. Example, you can say the ketoprofen, it is a drug of choice for sustained release formulation, but it has produced a gastric irritation during chronic therapy. Along with this, due to its low solubility, ketoprofen shows incomplete release from sustained release formulation. This problem can be successfully overcome when ketoprofen is presented in SEDDS formulation. This formulation enhances its viability due to increase the solubility of the drug and minimize the gastric irritation. Also, incorporation of gelling agent in SMDDS sustained the release of ketoprofen. Tipranavir and saquinavir SMDDS formulation have shown higher viability. Protection against biodegradation. Many drugs that are degraded in physiological system may be because of acidic pH in stomach, enzymatic degradation or hydrolytic degradation. Such drugs, when presented in the form of acidities, can be well protected against these degradation process, as liquid crystalline phase in acidities might be an act as barrier between degrading environment and the drug. SEDA formulation of ginkgo biloba was accordingly developed to increase dissolution rate and thus improve oral absorption and acquire the reproducible blood time profile of the active component of GBE. Slibin, the principal component of cardiosmoreous extract, is known to be very effective in protecting liver cells. The SEDDS formulation provides a greater increased level of in vivo viability of slibin, the level being at least four-fold higher than that achievable by conventional formulations. The marketed product of SEDDS